Good afternoon. My name is Don Tonti-Ploffel, and I'm the Associate Director of Academic Ventures and Engagement at Harvard Radcliffe Institute. At the start, let me acknowledge the members of the Radcliffe Institute Leadership Society and our annual donors tuned in this afternoon. Let me also specifically thank Ethel and David Jackson for their support of Radcliffe projects on the future of climate change. Your generosity keeps Radcliffe programming free and open to the public. We thank you. Today, it is my distinct honor to welcome you to this exciting discussion in the Institute's Climate Change Science Lecture Series, which is a key part of our multi-year focus on climate change and climate justice. This climate change initiative is our institute-wide and multidisciplinary effort to investigate the causes and consequences of the climate crisis, to develop strategies in adaptation and mitigation, and to address issues of climate justice in thought and in action, analytically, normatively, and sociopolitically. In particular, we seek to address all aspects of the climate crisis, especially as they bear on marginalized perspectives and vulnerable communities, locally, regionally, and globally. At the heart of this work is Radcliffe's commitment to supporting and sharing research that promotes innovative solutions and greater equity. This reflects Radcliffe's bedrock commitment to interdisciplinary study, informed by an equally deep dedication to inclusion. We firmly believe that these approaches will generate valuable new insights into effective and equitable ways of addressing climate change. Today's science lecture, titled Predicting Mosquito-Borne Disease Transmission in a Rapidly Changing World, to be delivered by our featured speaker, Professor Courtney Murdock of Cornell University, brings into focus the Institute's emphasis on addressing climate change from a multi-methodological, multidisciplinary perspective, particularly informed today by combining population-level macroanalysis with mechanistic models on the micro level. From climate change to land use, anthropogenic alterations to the environment are occurring at unprecedented rates and on an unthinkable scale. These developments alter the built landscape and our access to life-sustaining resources. They also disrupt the ecological relationships that bind together humans, mosquitoes, and parasites. Upsetting these relationships results in changes to the distribution of host organisms, rates of contact, the spread of infectious diseases, and the seasonality of transmission risk. What are the climate variables that influence mosquito-borne disease transmission? How can deploying advanced models of climate-based disease spread help to predict transmission patterns? How can these models help us to respond to the epidemiological effects of the climate crisis? Courtney Murdock is Associate Professor of Entomology in the College of Agriculture and Life Sciences at Cornell University, where she heads research on the ecological dynamics of vectors and parasites. Our discussant and moderator today is Professor Immaculata De Vivo, Faculty Co-Director of the Science Program at Harvard Radcliffe Institute, Professor of Medicine at Harvard Medical School, and Professor of Epidemiology at the Harvard T.H. Chan School of Public Health. On behalf of Professor De Vivo, I encourage the audience here to use the Q&A feature on Zoom to submit your questions at any time during the program. But please keep your questions concise so that we can explore as many questions and topics as possible. It is now my pleasure to give the virtual floor to Professor Courtney Murdock.
Thank you, Don. I'm very excited to be here today to talk about a topic that's very near and dear to my heart, and is also incredibly important, and to share with you some of the work that my group is doing currently. Okay, so today I'm going to start off by first posing a question. So the question is, what is the world's deadliest organism? Now, some of you probably realize that this talks about mosquito-borne disease, so you might be thinking it's the mosquito. And if that is the case, you are correct. So mosquitoes are actually the world's deadliest organism. And that's mainly because of the parasites and pathogens that they transmit to us. These diseases are responsible for killing 725,000 of us on average annually. Humans come in a close second. We're also very good at killing each other, which is a bit sad. And then some other animals that you often hear in the news as, taking out people like sharks, for example, actually contribute very minimally to reducing our numbers. And the reason why mosquitoes are so deadly is primarily because of this parasite that they transmit. So this parasite is human malaria. principally Plasmodium falciparum, and it's transmitted by mosquitoes in the species complex of Anopheles gambiae. Now, there are other mosquitoes too, such as Aedes species and Culex species that also transmit viruses to us that can make us pretty sick, but are not as deadly. And it's these pathogens and parasites that contribute to this overall mortality each year. Okay, so just to put some numbers on the effects of these parasites and pathogens. So for malaria, which I call a killer in the night, mainly because Anopheles comes into our homes and will feed on us overnight while we're sleeping. Historically, it's taken out about 1 million people annually. And unfortunately, this mostly is in children between the ages of 6 months and 2 years of age, and in sub-Saharan Africa. Now, we've made a lot of progress in controlling malaria and eliminating malaria from some countries through interventions such as bed nets to protect us from biting mosquitoes, artemisinin in combination drug therapy so that we can recover when we become infected, as well as indoor residual spraying of insecticides. But the total cost of these interventions annually is about $12 billion, so it's very expensive. And these interventions, while they've been successful, have stalled out in many countries. And then also malaria, along with other infectious diseases in these tropical countries, are significant contributors to poverty traps. So when you have a high burden of your population infected and sick and not working, it's really hard for these countries to improve their economies and also to pull themselves out of poverty. Now, I'm going to focus the rest of my talk on viruses transmitted by mosquitoes. And that's primarily because many of the most significant and recent invasions of emerging infectious diseases have been mosquito-borne and mosquito-borne transmitted viruses. And so what you can see here is a map of the world with various colors overlaid on top. And this is a map of habitat suitability for Aedes aegypti, which is the yellow fever mosquito, which is pictured in the left of this panel. So it's the brownish colored mosquito. And so areas that are red correspond to areas of high suitability, with cooler areas corresponding to lower suitability. And this mosquito is an invasive mosquito species that originally emerged out of Africa. And it was a tree hole breeding mosquito. So it used to be confined to the forest, but with increasing human encroachment, it's slowly domesticated to live in close proximity with us. So that now we provide the habitat and the blood meals for this mosquito. And as a result, it's moved around the world. And much of this distribution was originally set up hundreds of years ago due to European colonization and the slave trade. However, it's still being moved around by us today. And now it thrives quite happily in highly densely packed human environments, especially cities in the tropics where the environmental conditions are highly conducive. And this little mosquito here transmits some pretty nasty pathogens that can kill us and make us pretty sick. And so some of these include dengue fever. So dengue fever is also known as breakbone fever. It's not very pleasant. And it began emerging approximately 30 years ago. And now it affects roughly 400 million people annually, and it can cause significant mortality and can make us pretty sick. A more recent invasive mosquito-borne virus is chikungunya. So commonly it's called to contort or to bend up. So that's also not very pleasant when it comes to symptoms. It can cause fever like dengue, aches, and also rheumatoid arthritis symptoms. And so it was introduced into the Americas in 2013 and has proceeded to cause over 1.8 million cases in more than 44 countries and territories. So it can make us pretty sick. And then the most recent mosquito-borne virus that you probably are familiar with is Zika. So this was introduced into the Americas in 2015 and has rapidly expanded, causing thousands and thousands of suspected cases, and probably likely more because many of the cases were asymptomatic. It became really, it was pretty scary, mainly for its effect on pregnant women when they became, infected with this virus and some of the congenital and neurological defects that their offspring unfortunately experienced. And so, as I said previously, these mosquitoes tend to thrive in human environments, especially environments like this densely packed city. And so, to give you an idea of what the life cycle of a mosquito is, so a female mosquito will come in and she will lay her eggs along the water line of water containers that we use to store water. And when we fill these water containers, these eggs become submerged and they hatch out, and larvae hatch out of these eggs. Then mosquito larvae will go through 4 different stages of development and will eventually become a pupa. And then the adult mosquito, after 1 to 2 days, will emerge out of these, out of the pupal casing into the terrestrial environment. This female mosquito will then have to mate with a male, and she'll use his sperm to produce all of her offspring for the rest of her life. However, before she can take that, to lay her first clutch of eggs, she needs a blood meal. And because these mosquitoes are highly domesticated, they tend to live in our indoor environment. So they'll host seek on us throughout the day, and they'll rest, on the walls, for example, of this house here, under the draperies, or in the closet, where they will digest the blood meal and then develop their eggs to continue the cycle. So if she's carrying a parasite at this time or a pathogen at this time from her previous blood meal, she has the opportunity to now transmit this pathogen to members living within this home. Okay, so now if we step back and we think about the various factors that could influence the transmission of mosquito-borne diseases, these include environmental factors that mosquitoes experience in their habitat. So these will include things like abiotic factors like temperature, relative humidity, rainfall, as well as a suite of biotic factors like the quality and quantity of larval habitat and adult habitat, access to hosts for blood meals, competition within their aquatic environment with other larvae, predation, and even parasitism. And then there are also genetic factors that will influence their susceptibility and infectiousness for human pathogens that they can transmit to us. And human factors such as variation in immunity across populations, access to public health infrastructure, quality, integrity of people's homes, how much water is being stored, et cetera. And these factors all integrate to affect, the densities of mosquitoes that are present locally, their contact rates with humans, the probability of transmission of certain pathogens, and their distributions. And these will then in turn integrate to affect the global distribution of mosquito-borne disease. Okay, so now layer in the changes that we're applying in our local and global environment. So the synergistic effects of climate change and land use change, in particular urbanization, are two of the most defining environmental challenges of the 21st century. So these factors are altering the environment for mosquito-borne disease transmission rapidly. Cities are the fastest growing landscapes on the planet, and they're impacted by intensifying urban heat islands that then modify local climates that these mosquitoes experience. This causes mosquito populations to potentially expand in these urban environments. And this has led to explosive epidemics of key arboviruses like dengue and chikungunya in these tropical megacities that combine high human climates conducive to transmission. So, and as I said, currently 3.9 billion people globally are at risk of mosquito-borne arboviral diseases. And so what my research focuses on is, we try to understand the environmental relationships or the relationships that exist between key environmental factors and, mosquito and how these affect mosquito population dynamics and the transmission process. And we like to use mathematical models, which is basically a way for us to distill the complexity of this system to the factors that we think are most important for driving transmission. And then we explicitly define these relationships and these processes through mathematical equations. And then we use these models to predict, for example, how the number of cases of mosquito-borne disease fluctuate on a yearly basis and across the seasons. And then we can also do this across space to try to identify areas of the world or a city that might be at most risk. And then we can validate these models, these predictions from these models using actual case data. And when we have high-performing models, we can then do things like we can forecast the years and times of season that are most at risk for mosquito-borne disease transmission. We can predict the populations across different geographic regions or environments that are most at risk. We can take these models and then use them to anticipate the effects of future climate change and urbanization on risk. And then we can tweak various aspects of these models to see how different interventions might affect the total number of cases, for example. And then we can use this knowledge that we gain to target interventions to mitigate disease proactively. And so much of the work in my lab to date has focused largely on the effects of temperature on mosquito-borne diseases. And this is because mosquitoes are cold-blooded animals, so they cannot regulate their body temperatures like we can. And because of their small size, their internal temperatures quickly track and mirror temperatures in the ambient environment. And this has significant ramifications for the metabolic rates, how fast they develop, how well they survive and reproduce, their activity, and even how fast pathogens develop inside of them. And so this is kind of a complex slide, and I'm going to just basically highlight the things that I want you to take away from this. So we have panels here of mosquito and pathogen traits that are relevant for transmission. And we're showing how temperature affects these traits. And so we have traits, for example, that are important for governing mosquito population dynamics, such as EFD, which is the eggs females produce per day. We also have temperature relationships for the probability that larvae survive the larval environment. So that's PEA. We also have, mosquito development rate, which is MDR, so how fast they develop in the larval environment. And then we have adult lifespan, because that governs how much time a mosquito has to, take blood meals and to produce eggs, and how many offspring she will have across her lifespan. And then we have traits that are important for governing the transmission process, or the number of mosquitoes that are infectious, as well as the time that they are present, infectious and biting. So these include things like the biting rate, which is alpha. We have transmission probabilities that are associated with, for example, a susceptible mosquito feeding on an infectious host. What's the probability that she becomes infected, and vice versa? And then we have the extrinsic incubation rate, which is basically how fast it takes her after taking and imbibing an infectious blood meal to become infectious herself to transmit. And then what we can do is we use, so that's what all those traits are. And you can see the effect of temperature. And so what you can see is temperature has strong effects on all of these traits, and that these effects are nonlinear. So warmer does not necessarily equal better. And so for all of these traits, performance usually is optimized at or maximized at some intermediate temperature and constrained by a lower and higher temperature. Another thing to take away is that, the predicted thermal optimum and minimum and maximum for these traits are not the same across traits. And so what that means is the shape of these relationships also qualitatively differ. And all this means is that temperature has complex effects on the transmission process. But what we can do is we can take these temperature trait relationships once we've defined them, and we can incorporate them into simple models like what I have here, or more complex models to predict thermal suitability for disease transmission. So the model that I have here is just a mathematical expression for the basic reproductive number of a disease, or mosquito-borne disease. And basically what this means is that, how many cases would result from an infected mosquito upon introducing her, or an infected individual upon introducing this individual to a completely susceptible population. So if if the number of infected cases is above 1, for example, then you'd expect the disease to spread. And if it's below 1, you'd expect the disease to go extinct. And so what we can do is we can build these temperature trait relationships into this model. And in doing so, for dengue, for example, transmitted by Aedes aegypti here, you can see that the result of temperature on transmission is also nonlinear in this system. And this model would predict that dengue thermal suitability would actually be optimized around 29 degrees C. Okay, so today I'm going to tell you 2 stories. So the first is going to focus on defining the thermal niche for an emerging arbovirus, so for Zika virus. And this was conducted by a former graduate student in my group, Blanca Tesla. And then I have another story, which looks at how fine-scale variation in microclimate affects dengue transmission suitability. And this was conducted by several graduate students in my group. So Michelle Evans is no longer with me. She graduated, Nikki Solano, and Mike Newberry. Okay, so for the first story, so Zika emerged out of Africa in the recent past, but it almost feels like ancient history on this back end of the COVID pandemic. And it rapidly spread across the globe. So it was introduced into the Americas and Brazil in 2014, and then proceeded to spread rapidly throughout the Americas. And then this, coupled with an increasing number of infants born to Zika-exposed mothers with microcephaly and other neurological disorders, Zika was quickly declared an international public health crisis. And as a result, there was a huge initial push to map the global suitability and to predict areas of the world Zika could potentially emerge or become established in. And this resulted in several risk maps being published across many different journals. in the scientific literature. However, a general lack of data impedes our ability to predict accurately the transmission risk of emerging pathogens, mainly because we don't have these data yet since they're newly emerging. And so, this model framework, this is the same model framework I showed you previously, and is a temperature-dependent transmission model built for the dengue virus system. And so in particular, we lack temperature trait relationships for these infection probabilities, as well as for the extrinsic incubation rate for Zika virus. And so to compensate for this lack of data, this study and the previous studies I just showed you use temperature trait relationships for dengue virus, which is another Aedes transmitted flavivirus. That's not necessarily a bad assumption. They're both flaviviruses being transmitted by the same mosquito, and many other, aspects of these models, because they're transmitted by the same mosquito, hold. But we wanted to ask the question if this is a safe assumption. So is dengue virus a suitable surrogate for Zika virus? And is this generally a robust approach to take when data are lacking? Okay, so this is Blanca. So she set out to define the thermal relationships for these key transmission parameters. So vector competence are those transmission probabilities combined, and is basically the proportion of mosquitoes that go on to become infectious, the pathogen development rate, and then mosquito mortality. And then we updated that dengue temperature dependent R0 model, which I just showed you, with the Zika specific data. And then we compared the predicted temperature transmission relationships for dengue and Zika. And so this is, how we actually executed the experiment. So we went out and we collected mosquitoes from Chiapas, Mexico, from the field, Aedes aegypti. We brought them back into our lab and then reared them up so that they were at sufficient size to conduct these experiments. And then we took 4,000 female mosquitoes and we fed them a Zika infectious blood meal. And then we had 4,000 mosquitoes that we fed an uninfectious blood meal. Those are our controls. And then we distributed 2,000 within each of these groups across 8 different constant temperatures. And then every 3 days, we went in and we forced salivated mosquitoes, which sounds like torturing mosquitoes, and it kind of is. So we ended up, we did this across time because mosquitoes, when they take a blood meal that's infectious, they don't become instantaneously infectious themselves. So the virus has to get through, has to infect, replicate, and escape out of the midgut into the hemolymph, and then which is the hemocyl, which is the cavity of the mosquito, the body cavity of the mosquito. The hemolymph, which is the mosquito's blood, will then transport this virus to secondary tissues. The virus then needs to infect those tissues, replicate, and escape those tissues in order to end up into the saliva. And only at that point is the mosquito infectious to the human. So when we force salivate mosquitoes, we're looking for virus in the saliva. And we're trying to characterize when that happens across these different temperature treatments. And so to do this, we have to remove, the legs and wings from these mosquitoes because we don't want Zika infected mosquitoes flying around our insectary. That would be terrible. And then we insert their mouth parts into these pipette tips here, which have fetal bovine serum at the end. And when a mosquito imbibes that fetal bovine serum, she'll salivate. And if there's virus in that saliva, we'll collect it in these pipette tips. We then use something called plaque assays to determine if this saliva is actually positive for Zika virus. And plaque assays essentially are lawns of, a mammalian cell. So we use Vero cells. And when the virus actually lyses or kills the cell, it forms these plaques. And then you can identify if, a sample is positive or negative. And you can also count the plaques to get a sense of how much virus is in the saliva. And then we went in and counted the number of mosquitoes that died daily. And so we use this information to fit symmetric and asymmetric functions to between temperature and these trait responses. So for vector competence, which again is the average proportion of infectious mosquitoes that occurred for each temperature, you can see that a symmetric function fits best, and that we predict that the thermal optimum for vector competence would be about 30.6 degrees Celsius. For the extrinsic incubation rate, which is the average time it takes for a female to become infectious across each temperature, you can see we have an asymmetric relationship that fits best. and that the thermal optimum for this trait is actually predicted to be 36.4 degrees C based on our data. And then for lifespan, which is just the average number of days mosquitoes live across these temperature treatments, again, we have a symmetric relationship that seems to fit best, and we're predicting that the temperature optimum will be 24.2 degrees C. So again, we're getting different thermal optima and different thermal minima and maxima for each of these traits, which is interesting. Okay, so then we took these temperature trait relationships for Zika and we put them back into this dengue-dependent temperature mechanistic model. And we compared what these models would predict for thermal suitability for these two pathogens. And so you can see there are a lot of areas of overlap here. So our predictions for Zika are in dark blue. You can just pay attention to the middle lines. The dotted lines are the credible intervals or the amount of variation essentially around these relationships. And then dengue's our our dengue predictions are for our light blue or light blue colored line. And so what you can see here is that Zika virus had similar thermal optimum around 29 degrees C and thermal maximum as dengue virus. So that's predicted to be about 34 degrees C. And so what this means is models using dengue temperature trait relationships and to predict, areas that are suitable that are near the thermal optimum or warmer are probably doing a decent job at predicting thermal suitability. But where they break down is in these cooler environments. So you can see here that the thermal minimum predicted for Zika is actually 5 degrees warmer than what you would predict for dengue virus. And so this can have big consequences. So if we use both of these temperature R0 relationships and we project these relationships across space, we can do this by calculating, relative R0 at 0.1 degrees C increments using monthly mean current temperatures taken from the WorldClim data set. And warm colors correspond to increasing numbers of months that are suitable for transmission. So red areas correspond to like year-round transmission scenarios, suitability scenarios with more seasonal environments characterized by cooler colors. And so what you can see here is that, if you look at the estimated range with dengue data, we have much more suitability at northern latitudes and areas that are typically experience cooler mean temperatures than what you would expect if you use the models updated with the Zika temperature trait relationships. And so if you zero in on Florida, for example, Florida goes from having year-round suitability for the dengue using, the dengue data versus 6 months of suitability using the Zika data. And then we can also use the updated model to predict how thermal suitability is going to change with climate change. So this was done with Sadie Ryan and colleagues. And so you can use this to predict how thermal suitability will shift under modest warming scenarios and severe warming scenarios. And so this is this model would predict that 2.5 to 2.7 billion more people will be at risk with future climate warming. Okay, so to wrap up this story, so Zika virus environmental suitability we found is limited by cooler temperatures than dengue. And I think there's some interesting questions here about this is differences in the thermal sensitivity of flaviviruses, the mosquito vector, or both. With these, and we also show that climate warming will probably extend the thermal suitability for Zika virus transmission and enhance the overall risk of arbovirus exposure. And then to transition into my second story, climate and land use change likely will synergize to affect the microclimates mosquitoes experience and the environmental suitability for disease transmission. Okay, so we're going to step back. We're going to kind of narrow down from our continental predictions to finer scale predictions. And the reason why it's important to do that is because mosquito-borne disease transmission occurs at fine scales. Mosquitoes live in your backyards, and the transmission of disease and the scale at which we intervene also likely occurs at this level, mainly because mosquitoes do not disperse this far. And your backyard could look very different from my backyard, for example. So, your backyard could look like this urban slum habitat in an Indian megacity, or it could look like this village in Africa that consists largely of mud and thatch housing, or this rural scene from South Asia. And variation in these landscape features could result in different microclimates mosquitoes experience, as well as the availability of larval and adult habitat, This can in turn result in different mosquito abundances, human contact, and rates of transmission. And so understanding this fine scale of variation is important for identifying accurately, when the transmission season will begin and end, and identifying potential hotspots for transmission, both of which will be important for targeting interventions. And so we predict in these urban centers that urban landscapes are going to vary in microclimate, mainly because of variation in things like impervious surface and vegetation cover. So impervious surfaces are things like glass, metal, concrete. They tend to absorb heat slowly, or absorb heat throughout the day, and then radiate more slowly overnight. Impervious surfaces like vegetation and mud. And so due to increases in impervious surface and probably decreases in vegetation cover and highly built environments, we would predict urban environments to have higher average temperatures, so have urban heat island effects and lower relative humidity and urban dry island effects. And these panels over here is for a city that I work in Surat, India. And you can see here that, these highly urban areas correspond to warmer colors and warmer temperatures than the surrounding less urbanized landscapes. Okay, so for this story, though, I'm going to take you to the exotic location of Athens, Clarke County, which is in Georgia, in the vicinity of the University of Georgia, where I spent my time as an assistant professor. And we have Aedes albopictus, which is the Asian tiger mosquito here. It's another invasive mosquito, has similar ecology as Aedes aegypti, which I've been talking about in my other story. And it's a secondary vector for dengue and chikungunya and many other areas of the world. So the system's a nice system to ask some proof of concept questions about how microclimate might vary across urban landscapes. And so what we did is we took an impervious surface map of Athens, Clarke County, and we binned it into 3 different types of amounts of impervious surface. So these red areas correspond to high impervious surface sites. Blue areas correspond to intermediate impervious surface and more vegetation cover like the suburbs. And then these white areas correspond to rural areas with very low impervious surface. And then we went in and we selected 9 sites, 3 corresponding in each of these different land covers. And we did a variety of studies. So we did experimental studies where we put mosquitoes in pots, seeded with leaf infusion, and then we grew them across these sites that varied in microclimate. And we looked at things like the number of adults that emerged from these habitats, how fast it took them to emerge, their body size, because this is important for understanding how well they'll survive and reproduce. And then a subset of these adults we take back to the lab when we infected with dengue, because we were interested to know if there were any carryover effects from these microclimates that they're experiencing in the field on their ability to transmit dengue. We also did field sampling, where we put data loggers out into the environment across different impervious surface gradients and vegetation gradients to try to collect microclimate data. And we also did adult and larval mosquito surveys to look at how abundance is changing across these sites. And then we used a temperature-dependent dengue mechanistic model that you've seen to predict how environmental suitability will vary across land use and across seasons. And so the various data sets that go into this model are characterized by these orange boxes here. And we use these data to build, various models, which are in the green boxes, to make spatially gridded products, which are the blue boxes. And so, for example, we can use statistical models to build relationships between variation and land cover that we can get from satellite imagery and macroclimate with the microclimate data that we're collecting on the ground with data loggers. We can use this to build a microclimate model that we can then make microclimate maps. So, for example, predict how temperature might vary across Athens, Clarke County. We can do the same thing for mosquito abundance. So we can take different environmental variables, use statistical models to build relationships with abundance. And then we can make, spatial predictions for how mosquito densities might vary across Athens, Clarke County. And then we can go to the literature and get those temperature trait relationships that I've shown you previously. And we can use our, relative R0 model to infer how thermal suitability for transmission will then vary across Athens, Clarke County. And so this work was done in collaboration with Mike Wimberly. He's an excellent geospatial ecologist out of the University of Oklahoma. And we basically predicted how thermal suitability would vary across season in Athens, Clarke County. And so VC is basically the same thing as relative R0. And so warm colors correspond to high suitability and cooler colors correspond to low suitability. And what you can see here is, in the warm summer months, so June, July to August, September, suitability and high suitability seems to be more diffuse across the landscape. However, in the cooler months, when the temperatures start cooling off, like in September and October, thermal suitability contracts to these areas of high impervious surface where you have the urban heat island effect occurring. Okay, so to wrap this story up, so basically we found that microclimate varied significantly with season and land use. Urban sites in general were on average warmer. And despite the subtle variation, so in Athens, because it's a small city, we only had between our urban and rural sites maybe a 1 degree or 1.5 degree C difference in temperature. This variation did translate into differences in mosquito ecology and transmission potential in some of the experimental work that we have done. And I would imagine that these effects are probably going to be larger for some of these tropical megacities that I'm now conducting work in, because the temperature differential from urban core is much greater. We also found that the effect of land use on the environmental suitability for dengue transmission varied with time of season, which means that sites might vary in their environmental suitability and potential risk, as the season changes. And then if we can predict, where areas of high environmental suitability and risk occur spatially and temporally, we can potentially then target interventions to these populations most at risk. And so basically in the first story, what I want you to take away is, pathogens exhibit different physiological constraints, and these will have implications for their current and future global distributions. And they're not all the same. And then when we contract to smaller spatial scales, we see that seasonal and spatial variation in microclimate with land use can shape the temperature suitability for virus transmission. And the ultimate bottom line that I would like you to take away from this talk is, once we understand what these mechanistic relationships look like between environmental variables and key aspects of the transmission process, we can leverage this understanding to predict how environmental change will affect mosquito-borne disease risk now and in the future, with the hopes of being able to proactively respond to mitigate transmission. And that, I think, and I have to thank everybody who's helped me with this work. I have a lot of collaborators. This is not just me. The ones that are particularly important are highlighted, are bolded. I have to thank my funding and of course my current lab members. Now, I think it's time for questions. So I would like to bring Immaculata De Vivo to the virtual floor to join me in conversation and to help field audience questions.
Thank you, Courtney. That was absolutely terrific. There was a lot of information. And I actually want to thank you for bringing up the sharks early on, because we're so fixated with shark kills.
Yeah.
We've dedicated Shark Weeks on television. So it was an interesting to look at those statistics.
We should have mosquito weeks.
Clearly, we should have mosquito weeks just based on your numbers. So I'd like to have a short chat with you, as I like to say, before we take audience questions. And my first question is, so your talk, rightly so, as you said, focused mostly on how temperature affects mosquito-borne disease transmission, but obviously there must be other environmental factors. I would assume something like humidity, and influencing the transmission, not just now, but in the future. What would those be? And is your current work addressing these interactions?
Yeah, no, thanks. That's a good question. So yes, so as I alluded to in the beginning of my talk, there's a myriad of environmental factors that can influence the transmission process. We've started work with temperature because we suspect that probably has a huge effect. But no, there's going to be other factors. So one factor that you mentioned, relative humidity, is going to be important, and precipitation will be important. So precipitation is important in the sense that, depending on the mosquito species, if you have more rainfall and depending on where they develop, it can increase the amount of larval habitat that's present and can support larger populations of mosquitoes. What's interesting for the mosquitoes that I work on in urban environments, the link to precipitation is not as clear because these mosquitoes breed in water storage containers. So precipitation is going to affect water storage behavior as well as how fast the water is consumed in the water storage. And that's going to determine how much time larvae have to develop. So the relationship's not as clear for those systems and can be kind of counterintuitive. And then relative humidity is clearly is obviously important and is a next step. So if you think about, biological processes, metabolic rates are going to be defined by temperature, but the solvent for all of those reactions occur in water. So you need to optimize both the metabolic rates as well as, your water balance in order to, survive, reproduce, live. And temperature and relative humidity have this interesting interaction, and it affects the amount of water that the air can hold. So especially on the warm end, when temperatures are warm, the amount of water that the air can hold is high. And so what that means is there's a bigger difference between the outside environment and the inside environment for the organism that they must balance. And so they're subject to desiccation stress. And so what we're really looking at in some of my current work is trying to define those relationships between temperature and relative humidity and these traits that are important for transmission, see if that helps improve some of our predictions.
So just on that, because I find it very interesting. So as you mentioned, the temperature is nonlinear. So would you assume that humidity would also have a nonlinear?
Yeah, so some of the work that's been done in other systems, humidity, like being underhydrated and overhydrated, is not optimal. So it does suggest that it's a nonlinear relationship as well.
Yeah, that was. I'm sure there's going to be people in the Q&A who are also going to.
Yeah,
charts were pretty impressive. It's very clear that it's not linear. So
and then there are other variables. There's biotic factors too, like that are harder to get at, from a modeling perspective, just because they're harder to anticipate. They're less predictable across like a landscape. So like larval dense, competition with other larvae in the habitat can have a big impact on your transmission potential and the size that you come out. And so quality of larval habitat, amount of food that you have, these sorts of things are harder to integrate into models.
Right, but the temperature and humidity, to me anyway, would be something that one would focus with, the climate change, the temperature and climate change aspects of it. So I had another question. So the risk of transmission is also going to be affected by non-environmental factors, right? Which are going to vary with socioeconomic conditions. How does your research address those factors to move from environmental suitability to disease risk?
Yeah, so so there are obviously other factors involved. So you talked about some of them. So previously, most of my research is focused on environmental suitability, but we are now moving into trying to understand other aspects that would get us at risk. So these things can include, and I talked about them in the beginning of my talk, variation in immunity. You can have variation in housing structure that can increase access to mosquitoes and potential exposure. You have differences in water, access to piped water, for example, which can affect water storage behavior. Public health infrastructure access also varies. So what we're doing, so I have work ongoing in 2 cities in India, in Surat and Ahmedabad in the Gujarat region. And to get at that, we're basically doing socioeconomic household surveys across 2,000 households in each city, asking questions along those, along this vein to try to get at some of the socioeconomic and demographic predictors of risk. And then we're doing essentially what we've done in Athens in these 2 cities, where we're trying to get at environmental suitability, in particular how temperature and relative humidity affects environmental suitability. So then we want to layer these 2 things on top of each other to get at risk.
Right. Okay. And looks like there's a lot of questions coming in, but I want to just ask another question as it relates to prediction. So there are recent predictions, right, that malaria may decrease in Africa, while viruses like dengue and Zika could increase in prevalence with future climate change due to decreases in temperature suitability for malaria transmission and increases in suitability for these viruses. What are your thoughts on these future projections?
Yeah, So if we were just considering temperature and I guess focus, like current focus globally on interventions, then I think those are reasonable predictions. However, I think it's more complex than what that scenario might lay out. So the thought is that as climate change warms, suitability for malaria might decrease because the predicted thermal optimum for malaria is lower than for viruses, right? So while it's decreasing thermal suitability for malaria, it might be increasing thermal suitability for these viruses. And then we also have been focusing on virus control, mosquito-borne disease virus control in these countries. It's been all about malaria elimination. However, we have a new mosquito vector over in Africa, Anopheles stevensii, which is one I work on in South Asia. It's an urban vector, and it's spreading in cities. It was introduced into the Horn of Africa. And it has a higher thermal optimum. Malaria has a higher thermal optimum for transmission in this mosquito. And then also, we're not really thinking about how relative humidity might change. And so if things get wetter, in addition to warmer, it might buffer these mosquito populations. And so you might still have, enhanced transmission in these areas relative to areas that might dry out. So
Yeah, no,
complicated.
So climate change, right? So what are the current challenges in projecting future scenarios for mosquito-borne disease transmission with our changing climate?
Yeah, well, so some of the things I think about, so I'm not a climatologist, but some of the things I think about, one, we can build models to project, climate change scenarios, but it's really hard to validate those models, right? Because we're making projections 50 to 100 years in advance. So with near-time forecasting, we can make predictions, see how things play out, and validate those predictions, see how the model's performing. But with long-term forecasting, we can't do that. So that's a problem. And also, what happens in the in-between space is important. So like we can make near-time forecasting, with some accuracy between 1 to 5 years, and then we have these emission scenarios that project out 50 to 100 years. But what happens in the in-between could be really important for those longer-term scenarios and how they play out. And then there are other things I think about as well, but we can move on to make sure we have time for
the Q&A. Yeah, I have a bunch of questions I'd like to ask you, but unfortunately for me, I'm going to turn to the audience Q&As. We'd like to take a few questions. For those folks, just use the Q&A function at the bottom of your Zoom screen if you'd like to ask a question. But in the meantime, there are loads of questions here. So one question, Courtney, is, do all mosquitoes respond to temperatures as shown in your 7 graphs? Do some mosquitoes survive and reproduce at higher temperatures? Does that mean that those mosquitoes will outcompete the mosquitoes which expire or fail to reproduce at lower temperatures? If so, wouldn't global warming just promote the mosquitoes who don't mind higher temperatures?
Potentially. There's a lot there to unpack in that question. Yeah. That's an excellent question. Okay, so yes, I would say across insects in general and the mosquitoes that we've looked at, where we've characterized these temperature trait relationships, we see similar effects. However, mosquito species are going to vary in their thermal responses, and even I would argue even mosquito populations within a species might vary depending on where they exist in the world. So mosquitoes living in cool environments might have different thermal responses than mosquitoes living in warm environments, even if they're in the same species, right? So with climate change, yes, if a mosquito, if it moves into, if the temperatures move or the conditions move into situations that's not suitable for a given population or a given species, you would expect declines. And you could potentially have movement of other mosquito species or other mosquito populations, into those areas if they're more thermally tolerant of those temperature regimes.
Yeah. So there's another question. How do you have time to conduct many types of predictive modeling, experiments, and collecting of field data? I know the computers do a lot of the predictive work once you have the data, but there is still a lot of problem solving and troubleshooting to do.
That is true. It takes a long time. So that Zika experiment that I presented took us a year to run. So, to set up, troubleshoot, and run takes a long time. The field studies in Athens, occurred across, across 3 years. And they were running in parallel with each other because you have different people working on it. So it does take a long time. So from a mechanistic perspective, that's probably one of the weaknesses of this approach, actually, because it is fairly data intensive to generate these relationships in a timely fashion in order to respond. But I think when we're talking about climate change scenarios that are occurring later in the future, we might actually have time to do some of this mechanistic work. And then there are, pros and cons with all, many different ways of modeling these effects and projecting them into the future. So I prefer to work with mechanistic models because they fit nicely in with my experimental work, but you can build statistical models much more quickly based on current conditions to project into the future.
Yeah,
but those also have costs or
takes a long time.
Cons associated with those approaches as well.
Yeah, it does take a very long time, and the statistical models often, would benefit from empirical data that we need to collect. So there's another question about Zika. In 2015, 2016, Zika was most severe in northern Brazil rather than megacities such as Sao Paulo. And the question is why? That's
a good question. I don't know if we've resolved that question. So there's some suggestion that there might be some cross immunity, for example, that might be happening, with other viruses that are transmitted in that area, like dengue, that could be contributing to some of the effects that we observed in northern Brazil. Maybe population level differences, genetic differences, in how we respond to viruses. But yeah. I don't have a good answer to that question.
That's we have really good questions coming in, and I know I'm going to get a message soon to say one more question. So let me move on. How this model can predict the effect of long-term exposure to high temperatures? It says in the concept of global warming on mosquitoes and also on pathogens. I guess there's a missing word. How would this model?
Yeah, so basically, because we've gone in and we've measured and quantified what those temperature temperature trait relationships look like for the traits that are important for mosquitoes' ability to survive, reproduce, to bite, for their population dynamics. We've also done this for the pathogen, right? So we've looked at how fast it takes, how fast it takes for pathogens to develop and their ability to establish and transmit, and the effects of temperature. So we can incorporate those temperature trait relationships, those nonlinear functions, into these mathematical models to predict how changes in temperature in the future would, affect this transmission system.
Okay, so there's another question here, and then there's a three-part question that we should get to, but this question came in. How should we researchers consider the possible ecosystem-wide impact of intervening in the mosquito life cycle to control the infectious diseases?
Oh, that's a good question, because I'm an ecologist. So I like thinking about, connections and how tweaking one aspect of an ecosystem could affect another. I don't have a good answer to that question. So it depends on, I think it's an important question to ask, and one that we should be actively thinking about when we're trying to especially design interventions that are geared toward eradicating a mosquito species, either locally or globally. I think we need to think about, what the role of this mosquito species is in the surrounding community and ecosystem. That will unfortunately take probably a lot of work to try to discern, to see if it's redundant, if they're redundant and replaceable and don't have a big effect, or if they have an unintended huge effect that could shift the balance of the ecosystem in some fashion.
Okay, I think we have to take our last question. I'm going to choose this question. How do your models take viral mutation rates into consideration?
Yeah, they don't. That's a good question.
Oh, that's a good
I do have some work right now in collaboration with Colorado State University where we're looking at how temperature affects within mosquito, virus evolution. So there's, these viruses are kind of, they're sloppy replicators. So you can get a lot of variants that are produced throughout the course of the infection. So at least within the mosquito, where temperature really matters, we're looking at what comes in, in the different stages of the infection, how the community of viral variants is changing, or the population of viral variants is changing, and what comes out. So we're hoping to take that knowledge and use that to kind of address that very question. And we're doing this in the West Nile virus system with Culex tarsalis.
Oh, that's great. And unfortunately, I have to stop taking questions. And I want to thank you, Courtney. It was not just for today, but spending the last few days with me. It was fascinating talk. You're just so talented, and you're just so wonderful to work with. And with that, I want to bring it to Don, who is going to close us out. And I want to thank you again for taking the time to spend with us on a Wednesday afternoon. Don?
Great. Well, thank you. This concludes our program today. I want to thank Professor Courtney Murdock for her illuminating presentation, and both Professors Murdock and DeVivo for the expansive and provocative conversation, and you, our audience, for excellent questions and insights. Today's program has been recorded and will be posted on the Radcliffe website in about a week. For more information on future Radcliffe programs and to see videos of past events, please visit radcliffe.harvard.edu. Thank you again, and thank you for joining us today, and please take care.
Yeah, no, thank you very much. It was my pleasure.